This video is a recording of a virtual population lab. We will look at the population of two Paramecium species. We'll first set up test tube with each species by themselves, as well as a third test tube where two species are grown together. Then we'll monitor their population density over a period of 16 days. On this test tube here, I have set up a population of Paramecium aurelia culture only. In the middle test tube, I have set up a test tube of Paramecium caudatum culture only. And in the third test tube on the far right, I have a mixture of Paramecium aurelia cultures and Paramecium caudatum cultures. Now let's look at what happens on day 0. We look at the slides for Paramecium aurelia, and what we see, there's only one cell per milliliter on day 0. For our second test tube, we also see there's one Paramecium caudatum cell per milliliter in our second test tube. And in our third test tube, which is the mixed culture, we see there's one cell of Paramecium aurelia and one cell per milliliter of Paramecium caudatum. Pause the video now and record the information. Let's continue. We'll clear off slides, advance by 2 days, obtain clean slides, and take clean samples. In our first test tube, we now see there are 6 Paramecium aurelia culture cells per milliliter when grown individually. There are also 6 cells of Paramecium caudatum per milliliter when grown individually. And now we're going to pause the video so that you can count the number of Paramecium aurelia cells and the Paramecium caudata cells per milliliter. We now clear the slides, advance by 2 days, and take the samples again. Here is the culture of Paramecium aurelia when grown alone. Pause the video to count the number of Paramecium aurelia cells per milliliter. Now we see the number of cells per Paramecium caudatum. Again, pause the video to count the number of Paramecium caudatum cells per milliliter. And our third test tube is a mixture of both Paramecium caudatum and Paramecium aurelia. Once again, pause the video to count the number of cells per milliliter of each species. Let's advance to day 6. Let's advance to day 8. Let's advance by 2 more days. Let's move on to day 12. Day 14. And now our last day, day 16. Now that you have determined the population of each Paramecium species per milliliter when they're grown alone and individually, continue your lab graphing the data on your graph paper.